Good morning students, welcome to the class. Today we are moving on to the second part of the chapter, plant growth and development. Today we are going to discuss about plant growth regulators. Okay. So the topic for today is plant growth regulators, and few more points we will be discussing. Okay. So this plant growth regulators, if you are seeing, there are two types of plant growth regulator. One is your plant promoter and another one is your plant growth inhibitor. So one is helping in promoting the growth of the plant and other one is inhibiting the growth of the plant, means they are not allowing the plant to grow, rather than they are carrying out some other functions. Okay. So that is growth inhibitors. What do we call them? They are inhibiting, they are stopping, they are restricting. Okay. So that is growth inhibitors. Now plant growth promoters, or when you are seeing those, the hormones which are helping in the growth of the plant are of three types. So what are they? One is auxins, second one is cytokinin, and third one is gibberellins. So plant growth regulators are otherwise called as plant hormones. Okay, like we have hormones, insulin we learned, like that. Plant are also having hormones. These are the hormones, right? So they can be called as plant growth regulators, or it can be called as hormones. Phytohormones can also be called phyto because it is related to plants. Okay. So growth promoters are auxins, cytokinin and gibberellins, and growth inhibitors are ABA, that is abscisic acid, and ethylene, a gas. Okay. So ABA and ethylene produced by the plant, when you are seeing, they are acting as growth inhibitors. Today what we are going to do is that we are going to learn each and every plant hormones. Okay. So we are going to learn these hormones. The first plant hormone which we are going to discuss is auxins. So plant growth regulators, when you are seeing promoters, under that the first one was auxin, second one was gibberellins, third one was cytokinin. You can change the order, doesn't matter. Now moving on to this auxin. So auxins, when you are seeing, what is this auxin doing? I will just change the color. Yeah, fine. Auxins, how it was discovered? It was discovered by these two persons. One is Charles Darwin and another one is Francis Darwin. From where did they isolate auxins? Where did they got auxins? They got the auxins from the tip. Okay, tip means you can understand the end parts. Okay, so in which, so if you are taking, this is a seed. Yes, so inside the seed there is an embryo. This embryo, when you are seeing, the embryo is protected by two coverings, we have learned, isn't it? One is the covering of the root, which is called as the coleorhiza, and another one is the covering of the shoot, which is called as what? The coleoptile. So and the tip of the coleoptile, means the embryo tip, the coleoptile, when the embryo is slowly growing, you can find the coleoptile is also a bit growing. So in the tip of the coleoptile, from there only what was discovered, auxin was discovered, means auxin was isolated, they got auxins from them. Okay. So Charles Darwin and Francis Darwin, they isolated auxins from where? From the coleoptiles of oats, you know, seedlings. So this oat seedlings, from there, who isolated? It was discovered by these people, but isolation was done by whom? This FW Went. Okay. So they isolated it as what? AAA, indole acetic acid, and indole butyric acid, IBA, IAA and IBA were isolated from where? Oat seedling, and this were termed as, discovered as auxin by whom? These two person. Okay. So Went isolated it and it was discovered as auxins, gave the name as auxins by whom? These two persons. Okay. Now what is the effects? Effects means what is the thing, what is this auxin doing in plants? Okay. What are its functions, what are its uses, what is the significance or the importance? Now when you are looking into the effects, they initiate rooting in stem cuttings and plant propagation, helps in propagation. Propagation means growth. Initiate rooting in stem cutting means, like for example, this is a rose plant. Imagine if I am cutting a rose plant and putting it in a soil which is containing auxin, okay, where I have put some auxin into the water. With the water I have mixed auxin and I have putting it in, or in the plant, just like that, if I am keeping it in the soil, auxin is already present in the plant, isn't it? So this auxin hormone, what they do is that they initiate the rooting. You can find after some times the roots would have developed in that plant, isn't it? So how is this roots coming? The roots are initiated by some mechanism, chemical mechanism. Who is playing a major role there? The auxins are playing a major role. So, they are initiating rooting in stem cutting and thereby helping in the propagation of the plant, growth of the plant. Next is they are promoting flowering. So, to produce flowers also they are playing a major role. Auxin, when it is produced, then only flowering is possible, and they do not allow the fruit or the leaf to drop very fastly, means they do not allow them to grow old very fastly. If this auxin is present, they will remain young for a longer duration of time. Okay. And promote abscission of older mature leaves. Abscission means what? Two terms you have to learn. One is senescence and another one is abscission. Okay. So, what is senescence means? Aging. Aging is called as what? Senescence. The plant, some of the parts of the plants, the cells are becoming old and they are dying. So, that one we call it as what? Senescence. So, these aged senesced part, when you are seeing, either it is a leaf, yellowing has taken place, the leaf has lost its lifespan, it is dead now. What happens? This is senescence. It has to fall down. That falling down, dropping down, okay, so that it is detached from the plant, so that the new parts can take up the role of the one which has died, isn't it? So, that dropping or that falling of these leaves, we call it as what? Abscission. Leaves of fruits or flowers, when they become old, their lifespan is over, they fall down. That process we call it as what? We call it as abscission. And what is senescence? It is aging of the parts of a plant. Okay. Yeah. Now, when you are seeing this auxins, they are helping in promoting abscission of older mature leaves. So, older mature leaves and all, they are detaching them from the plants. Okay. So, drooping is taking place. Next, uses. What are the uses of auxin? It is inducing parthenocarpy. One new term, what is parthenocarpy? Parthenocarpy means producing seedless varieties. You have seedless apple, you have seedless orange, you have seedless grapes, right? So, if auxins are artificially put in these plants, they induce what? Parthenocarpy. What is parthenocarpy? You have learned, would have come across this term, isn't it? So, parthenocarpy means developing seedless varieties. Okay. Next, one of the auxin named as 2,4-diethylamide, 2,4-D. 2,4-diethylamide, in short we call it as 2,4-D. It is used as a good weedicide, herbicide. Herbicide means, you know, it is helping in killing the unwanted plants. So, weedicides, herbicides we used to apply in the fields, right. So, this is auxin is one of the weedicide which can act as what? A killer for weeds. Who is that? What is that auxin? That auxin's name is 2,4-D. 2,4-D is acting as a weedicide or a herbicide. Very important question. Okay. So, 2,4-D is coming under auxin and they are acting as a herbicide or a weedicide. So, they are killing what? Dicotyledons weed. Dicots, you know, dicot weeds have been killed if we are spraying 2,4-D onto the lawn, okay, grass lawn or the field you are spraying it. So, prepare. So, since they are killing the weeds, weed free lawns we can get, okay, weeds will not grow there. So, that is a very good advantage for the gardens to look very beautiful. Control xylem differentiation, that is the next point. Control xylem differentiation means xylem, when you see, they are complex permanent tissue, right. So, xylem cells have to be different from one another, like xylem tracheus is different from xylem vessel, is different from xylem parenchyma, is different from xylem fibers. So, all this differentiation is taking place. Xylems are differentiating into different types of cells because of this auxins. Okay. Other than that, they also help in what? The cell division. So, these are the different uses of auxin. Now, growing apical bud, very important. Auxins, they play a major role in apical dominance. Apical dominance means if auxin is present, the tips keeps on growing, the apex keeps on growing. Okay. They do not allow the formation of the lateral parts. The apex, the tip grows, okay. So, apical dominance is one of the property of what? This auxin. So, growing apical bud inhibits the growth of lateral buds. So, if at all this auxin is present in high quantity, they will grow much longer. Sidewise you cannot find the lateral buds are coming and you cannot find the flowers are coming from sides. So if you are cutting this, so in between they used to tell, right, roses or whatever it is, the plants you have to cut so that you are getting the lateral growth. Why it is said that you have to cut the tips? Because the auxins are highly concentrated on the tip, and if this concentration auxins are present on the tip, then the plant will be growing tall, rather they will not give rise to branches because of the presence of this auxins. So in order to prevent that, we used to cut the tips. Okay. So auxins play a major role in apical dominance. Okay. So removal of shoot tips. So if I am removing the shoot tips, results in the growth of what? The lateral buds. Okay. So you want the plant to grow in the lateral way much. It should be leafy much on the lateral sides. Lateral means what? On the sides. Then you have to cut in between the plants, the tips of the shoot. Okay. So that is regarding auxin. And this is the example, rooting has been given, how they are producing the rooting. That is the major property of what? This auxin. Now gibberellins. Gibberellins is the second plant hormone which we are going to discuss. It was discovered by Kurosawa. He identified gibberellins in a fungal pathogen. Okay, he identified a chemical compound called as what? The gibberellins. In where? In a fungus, Gibberella fujicorii. Under that only he identified this gibberellins were present. How he identified, how the gibberellins, how did he identify? See, this Kurosawa and his colleagues were studying what? The plants suffering from a disease called as Bacchain disease. Okay. So plants were suffering from a disease. What is the name of the disease? The Bacchain disease. So this disease, they were studying why the disease is caused. Okay. So when they learned what is the symptoms of this disease, they found it as foolish shading. Foolish shading means the plant, they will be growing very long. The internodes and all, when you see, internodes means what? The nodal regions where the leaves arise, that one we, or the branch arise, we call it as what? The nodes. So if this Bacchain disease is attacked, what is happening is that the plant is growing very tall. Why it is growing very tall? Because the internodes, when you are seeing, the distance between the internodes are increased. Previously, imagine it was like this. If this disease is caused, what happens is that the distance between the internodes increases. Okay, so here the branch, here the branch, here nearby it was, but here it has gone apart, which means the plant internodes are increasing, and henceforth what is happening? They are increasing in the lengthwise. Lengthwise, when you see, they are increasing a lot. Okay. So this disease was noted in rice. Which variety? Rice variety. So this disease, when was studied, it was caused by a fungus. They learned that it was caused by a fungus, and name of the fungus was this. And they are helping in what? Cell elongation and cell division. So this compounds which was secreted by this fungus, what this fungus was secreting, that chemical compound, that is the gibberellins, which was isolated from this fungus, was sprayed onto uninfected plants. So imagine these are affected plants, from them he isolated the chemical which is responsible for the disease. So the fungus they isolated, from there he isolated gibberellins, and this gibberellins were sprayed over which one? They was sprayed over the uninfected plants. What happened? Immediately you can find the uninfected plants also started growing long, which means that chemical compound is having the property of increasing the internodes. Okay. So that is the property of what? This gibberellins. So gibberellins or gibberellic acid, since it is the name itself, it signifies they are acidic in nature. Okay. That is number one. They increase the length and cause fruits to elongate and improve its shape. So fruits are elongating. Yeah, like grapes, when you are seeing small round, in that case they are becoming bigger. So elongation is taking place. So the shape is improving. Okay, pineapple, it was small, now it has increased. Means obviously if the length increases, the quantity increases, the weight increases, the yield will be more, isn't it? Delay senescence. Delay senescence means was senescence, you know, death, isn't it? So they are delaying the senescence, another property. That's why if the death is means they are not ripening very fastly, they are not dying very fast, they are not decomposed very fastly, obviously the market period will be more, isn't it? They will be lasting in the market for long. And gibberellic acid 3, many are there, many gibberellic acid, 1, 2, 3, like that, many are there. Gibberellic acid 3 is used to speed up malting process. What do you mean by malting? Malting means what we used to do is that these grains, rice, wheat, barley, or whatever it is, the grains are collected, the grains are steepened, thereafter it is dried much, and it is used for producing malt. Malt, you know, like drying, making it into powder, thereafter crushing it very nicely, and it is used for making for brewers, malting, you know, for making beer and all, whiskey and all, they are using it as a raw material. So, that process we call it as what? Malting, in which malt is produced, and that malt is used for fermentation process in order to yield alcohol. Okay. So, for brewing industries, they are playing a major role, this gibberellic acid, as they help in what? Malting process. Okay. And uses. So, when you see this gibberellic acid, if this is the sugar cane, okay, it was having internodes like this. In this sugar cane, if I spray gibberellic acid, what will happen? The internode will increase, isn't it? So, what will happen to the plant? Obviously, you can find the length of the internodes will be more. So, the yield will be more. You are having much sugar cane, the nodes are less. Close by, they are far apart, so they are much longer, the size will be good. Okay. So, that is another point. Fastens maturity period, I told you, they will be fastening up. Maturity, it will be fastened up, means they will grow older faster, but it will not die soon. Okay. That is another one. Promotes bolting. Bolting means what I told you, increase, that only. In sugarcane I told. Bolting means what? In sugar canes or in cabbages, the internodes length is increased. So, automatically the leaves will be coming far apart. So, the size of the cabbage or size of the stem of the sugar cane will be much good. You can see this is again the grapes. Internodes are far apart, so automatically the quantity will be more. This is a cabbage, you can find how big they are, isn't it? If the internodes are very close by, then you can find the cabbage is smaller in size. If the internodes are far apart, then the cabbage will be somewhat longer in size and the size will vary. Okay. So, these are the differences between what? This, this was previous, applied, you can find the size internode has increased. So, here they are closer, here they are far away, so the length is more. So, that is the property of what? The gibberellins. The next hormone what we are going to learn is cytokinin. It was discovered by Zug and Miller. It was isolated in the form of a crystals, okay, and when they isolated this, which one? This cytokinin, it was from this coconut milk, corn kernels and all, they isolated this active substances, and he named it as was kinetin. It was named as what? Kinetin. Later it was named as what? Cytokinin. Now, what is the effects of this cytokinin? What is the role of cytokinin hormone in the plants? They are synthesized where rapid cell division takes place. So, wherever the rapid cell division takes place, obviously it will be meristematic region. So, in the meristems, lateral as well as apical meristems, you can find what? This cytokinin is produced. And they produce new leaves, chloroplasts, they help in the synthesis of chloroplasts, lateral shoot growth. I told you auxin is helping in what? The apical dominance, they will grow lengthwise, but when you are applying cytokinin, the lateral growth will be more, because cytokinin helps in what? The development of the lateral growth. So, you can find if you are applying cytokinin artificially to a plant, then what will happen is that this plant will be giving rise to many lateral branches, lateral adventitious. This one we call it as what? Adventitious. Sidewise you can find many are developing. Okay. So, like this they are helping in what? Lateral shoot growth or adventitious shoot formation is characteristic feature of what? The cytokinin. Uses, it overcomes apical dominance. So, if you are applying the cytokinin, so the apical dominance is stopped. So, the plants will grow on the lateral side more than on the apical side. Promote nutrient mobilization, means cytokinin, if you are applying, the nutrients will dissolve very nicely in the water and they will reach each and every part of the plant, and this nutrients will delay what? The leaf senescence, means the death of the leaf will be delayed in case cytokinin is applied, because they provide minerals to each and every part of the plant and they help in what? making the plant stay for a longer duration. Okay, this leaves all the cells stay for a longer duration. So that is the role of what? The cytokinin. The next hormone what we are going to learn is ethylene. It is a gaseous hormone. You have to remember, one of the gaseous hormone this present in plant is what? The ethylene. This ethylene was discovered by Cousins. So H.H. Cousin was the one. He said that how he confirmed that ethylene is produced by plants. He kept ripened orange. Ripened, you know, ripened means somewhat it's emitting some sort of gas. Okay. So this ripened orange was kept with unripe bananas. So he kept many. So here imagine this is unripe bananas were kept here. In that he kept a ripened orange. Okay. After some time what happened, he saw that all these bananas got ripened, which means this orange is producing some volatile substance, okay, so which was gaseous in nature, and this gas enhanced the ripening of what? This banana. So like that he understood that some gas is eliminated from them. When he studied, he understood it was ethylene gas. Okay. So ethylene is a gaseous hormone. What is the function? Promotes, what is the effects if you are saying? It promotes senescence. Senescence, you know, so it is an inhibitor. So obviously they will help in death of the plant faster, as well as abscission, means dropping. After they die from the plant, they are detached. So that is abscission. Highly effective in fruit ripening. So fruit will become ripened very fast. Okay. Enhances the respiration rate. So respiration rate of the plant will increase if you are applying this ethylene. Seed and bud dormancy is broken. Dormant means what? A seed. If you are taking this, like for example, if you are taking any pulse, okay. So if you have taken a pulse, and this pulse, if you are seeing, you are keeping like this pulse, if you are keeping it just like that, it can stay for a longer duration, isn't it? Green peas or dried one, whichever pulse you are taking and you are keeping it for a longer duration, it will be like that only. It will not germinate. But they are alive. They are alive because you know they are dormant. Dormant means what? They are resting. Okay. So when you take the seed and take this pulse and put it in the soil, and you apply water, airy place, sunlight, everything is available, you can find what will happen. Or overnight also, if you are just keeping it in the water, you can find the seed will swell up, and thereafter after you are keeping it for long, what will happen? The seed will start to germinate, isn't it? So this being in inactive form, dormant form, we call it as what? Dormancy. Okay. Unless the favorable condition comes, water availability, oxygen, everything comes, the seeds can remain dormant. After that, when they are getting favorable condition, the seeds will germinate. Okay. So when you are seeing this ethylene gas, it is breaking what? If you are applying this ethylene over the seed or the bud, the dormancy period will be broken and they will germinate very fast. Okay. Initiates germination in peanut seeds. Sprouting tubers. So potato also, ethylene gas is exposed, means the tubers, when you are seeing from the potato, you can find the leaves are starting to come. Promotes root hair and root growth, root hair formation and root growth, they are helping in. So this is the effects of what? This ethylene. Used to initiate flowering. So if you are applying ethylene, flowering will take place from, obviously flowering, if it is taking place, it is helping in synchronizing for fruit. So flowers are formed, obviously fruit will be formed. So induces flowering, regulates what? All the physiological processes. Physiological means functional part of the plant is regulated fast. Every process will be taking out fast. Since the respiration is taking out fast, all other processes will also be simultaneously becoming faster. Okay. Hastens fruit ripening, means it increases. Hastens means what? It increases the fruit ripening. Accelerates abscission. Abscission means, you know, the dead parts are made to fall down, and they help in the growth of the female flower. Female flower, they promote the growth much faster. Okay. So this is regarding your ethylene. Now next is your last plant hormone called as what? Abscisic acid. This abscisic acid was discovered by many researchers in some in different substances, different plants. And when learning on to this effects, what are the effects? They are regulating abscission and dormancy. Okay. They are regulating abscission and dormancy, means how abscission is being controlled, dormant period is being controlled by this abscisic acid. It stimulates. So, this abscisic acid is otherwise called as a stress hormone. Listen very carefully. Why it is called as a stress hormone? Listen, it is having a good role also, this abscisic acid. Now, I am having a plant, okay. This plant, now it is summer and I am not applying water to it. So, what will happen if the water is not applied? After sometimes you can find because of deficit of water, the plant can die, isn't it? Because it is summer and the water is not available artificially, we are not providing also. So, automatically what will happen is that this plant has the ability to die. But in order to increase their lifespan, this hormone plays a major role. How? Listen carefully. Availability of water is very less, as I am not applying it. What is there is only there. For some days I am not applying, maybe for a week I am not applying. So, within a week the plant can die, isn't it? But the plant is not dying. Why? Because this abscisic acid, what it is doing is that they are helping in the closure of the stomata. So, in the leaves what is present? The stomata is present, the stomata is closed. So, when the stomata is closed, do you think the transpiration, the water loss from the leaves will be taking place? No, the water will not be lost. So, water is not lost, transpiration is not taking place. So, water which is given out by the leaves by the plant is very, very less, isn't it? So, the plant is not giving water, they are taking very less amount of water and trying to adjust to the water and they are staying alive, okay. So, this property is only because of the presence of water, this abscisic acid. Imagine if this abscisic acid is not there, the transpiration is taking place, then what will happen? The plant will die very fastly, isn't it? So, they are reducing the transpiration, thereby how much water is present, they are utilizing it for themselves and trying to accommodate to the stressful environment. What is the stress there? Unavailability of water, okay. That is why we call them as what? The stress hormone. So, name the stress hormone in plant, it is abscisic acid, okay. So, stimulates the closure of stomata, increases tolerance, yeah, state development, they are helping in maturation, they are helping in dormancy period, okay. They are increasing the dormancy period. So, stressful environment, drought it is, imagine it is very drought now, the seeds are not getting any favorable conditions, but they are remaining alive because of the presence of water, this abscisic acid, okay. So, without desiccation, desiccation means what? Without any injury, the seeds are safe because of the presence of what? This abscisic acid. Now, uses, there are number of events in a plant where more than one plant growth regulator interact. So, you can find all these means dormancy, thereafter abscission, senescence, apical dominance, all these are the functions of a plant. And all these hormones together play a major role, okay. Based on the environmental condition, which is more, which is less is being determined. Based on that, you can find the plant is getting adapted to the things and the hormones are synthesized based on the requirements, and you can find the plant is staying alive as well as they are ripening, they are giving fruits, they are senescence is taking place, abscission is taking place, and the role continues. So, this is regarding your abscisic acid. Now, listen, a new term what we are going to discuss today is another one is photoperiodism. What do you mean by photoperiodism? Photoperiodism is something related to flowering, okay. So, a plant will produce flower only if the plant is exposed to light for particular duration of time. If the plant is not exposed to light, then they will not produce any flowers, which means for flowering light is playing a major role, okay. Now, listen carefully, so you would have understood the term photoperiodism. Photo means light, periodism means for a particular period. Now, this plant is exposed to light, only just exposure to light for 1 minute is enough, or only exposure to light for 1 hour is enough? No, it is not like that. It is having some particular duration, that is why it is period, okay. So, it has to be exposed for a particular period. Based on that, there are different types of plants. Okay, how much the light has to be exposed for how much duration, based on that there are different types of plants. So what is the different types of plants if you are taking? One is your long day plants. As the name suggests, long day. They have to get exposed to light for a longer duration. Only during summer, wherein you can find the days are long, that time only they are active, they will be flowering, other times they will not. Another one is short day plants. Short day plants means they should not be given light for a longer duration. They need light only for a shorter duration. If more is given, they will not be flowering. Okay. So, for a particular short duration only they need. Another one is your day neutral plant, which means light is needed, but duration is not a matter. Light is needed for some duration. Okay, in some duration means it is not rigid that very less is given or very long is given, like annual flower. Every time you can find a plant having flowers, producing flowers, means they are day neutral. It doesn't matter whether it is long day or short day. Light is there, okay, fine, I will flower. Okay, but long day they need long duration of light, and short day they need what? Short duration of light. Based on that, examples are given. Now, when you are seeing long day plants, example is radish, sugar beet and all are long day plants, they require light for a long duration, then only they will be growing and flowering. Okay, and short day plants, they require exposure to light only for less than critical duration. Then the critical duration, critical means what? This much amount of light is needed, that is optimum, means that is needed. Below that, what is not possible? Flowering is not possible. So, that one we call it as what? The critical duration. So, this critical duration, less than the critical duration is enough for flowering, which means that it is short day plants. So, example tobacco, chrysanthemum are all short day plants. Day neutral plants, flowering not induced by duration of light, like tomato and cotton and all, they doesn't matter at all. They need light, that's it, duration doesn't matter. Then we call it as what? Day neutral plants. The last one what we are going to see is that you can find short day plants, day length less than 12 hours, long day plants, day length more than 12 hours, here you can find, and day neutral means what? Day length it is not at all immaterial, means it is not at all a factor. Okay, so that is what? Day neutral plants. One more term what we are going to discuss last is your vernalization. So, with this the chapter comes to an end. Vernalization. What is vernalization? Listen carefully. Vernalization means flowering is possible only if a plant is exposed to very low temperature. Okay. If this low temperature is not provided to the plant, so imagine this is plant A. So, this is what? This is plant A. And if this plant is not given what is that? Temperature, means low temperature, it is not exposed to low temperature, then the flower will never be produced. Okay, what they are not going to produce? Flower is not going to produce, which means low temperature or critical low temperature, critical low temperature is responsible for the growth of the flower. Flower will be produced only when they are exposed to low temperature. Vernalization refers to the promotion of flowering by a period of very low temperature. Okay. The plants for which flowering is either quantitatively or quantitatively, means quality of light is also important, quality of temperature is also important. Okay. Dependent on exposures to low temperature. Quality means what? Quality of the flower or quantity of the flower, how much it is going to produce flowers, and the quality, whether it is going to produce a nice one or not that healthy one, it all depends upon what? The low temperature. Then that process we call it as what? Vernalization. Okay. So, it prevents precocious reproductive development, means what? In the beginning itself, before maturation, if the plant is becoming matured. Imagine, means for example, it is seen that the plant will mature only after 6 months, but the plant is all of a sudden maturing at 3 months, means it's not that good. It is not that good in some sense. Sometimes it can be good also, but in some flowering plants when you are seeing, it's not that good. Okay. So, in order to prevent that one late in the growing seasons, so what we are doing is that we are having what? We have to give them some low temperature, only then the plant are maturing, then it will be a good growth qualitatively as well as quantitatively, the flower will be good. Okay. So, enables the plant to have sufficient time to reach maturity. So, for the plant to reach maturity, we have to give them time. They should not flower all of a sudden. They should flower only after a particular duration. Then only you can find around the year you can find that crop is available. Okay. Only during this time the crop is available, afterwards we will not get it. No, nowadays you can find the varieties which are developed are means the fruits or whatever it is, they are available throughout the year. You can get it. Maybe the amount is fluctuating. When it is seasonal, you will get it in low cost, but when it is not seasonal, you will get it in high cost. But the plants are there, isn't it? The vegetables or fruits are available. So, example, wheat, barley have two kinds of variety. You can find one is the winter variety and another one is the spring variety of wheat or barley. Okay. So, spring variety, when you are seeing, they are planted during spring. When it is planted during spring, and they come to flower and producing grain by the end of the growing season, by the end of this spring season, when you are finding, they are flowering, right. Another one is your winter variety. Winter variety, if you are putting it in the spring, then what will not happen? Flowering will not take place. So, what you have to do, winter varieties has to be planted in autumn. So, when I have to plant it, I have to plant it in autumn. So, you can find in spring they are very small, growth only will be taking place, small seedlings will be coming in spring, and around midsummer, when it comes to around midsummer, they will, winter is over, they have got a temperature, low temperature, then when it comes to summer, you can find what will happen, they will be producing the flowers. Okay. So, what is vernalization? Only the definition is more important. So, vernalization is the plant will flower or the plant is promoting flowering only when exposed to low temperature, we call it as what? Vernalization. With this, we are coming to the end of the chapter. Today we have discussed what? We have discussed about plant growth regulators, that is plant hormones, phytohormones, who were they? Growth promoters and inhibitors. Promoters were auxins, cytokinins and gibberellins. Inhibitors was abscisic acid and ethylene. And then we discussed about photoperiodism, and thereafter we ended with vernalization. With this, the chapter comes to an end. Hope you would have understood, and plant physiology was over. Next, we will be moving on to our human physiology, that is functions of our body, different systems we are going to learn. Okay. Thank you.